Hello and welcome to my Montana Western Pearl Shell talk. I am David Stagliano and I've been working on the freshwater mussels in Montana since I got there back in 2003. For me, this project has been a culmination of many years of observational data gathering on this underappreciated group of invertebrates, and we are moving towards more conservation guided actions on this species. The funding for this research has come largely through state wildlife grants, from the U.S. Fish and Wildlife Service through Fish, Wildlife, and Parks, also from American Fisheries Raft grants, Western Division Small grants, and the support of the Montana Heritage Program through the University of Montana. And I also cannot thank my co-authors enough, Michelle Anderson and Kristen Cook. I am so happy to be giving this talk to a bunch of mussel-heads so that I do not have to talk about the difference in reproductive strategy of our unionid mussels. Of course, our true freshwater mussels rely on a host fish and oftentimes a specific species of host fish for the metamorphose of larvae as well as dispersal of juveniles. And this sets up our mussels at multiple avenues to limit recruitment, basically from juvenile to that subadult. You could have silt, you could have the loss of host fish, sedimentation, all sorts of factors. But this study is going to evaluate some of the limiting factors of the life histories of the pearl shell that are causing a significant decline across Montana. Back in 2008, we developed viability classes to describe our Montana pearl shell populations based on NatureServe criteria. Viable populations have to have some recent recruitment within the last decade or 30 millimeter mussels to be considered viable. You can have hundreds of individuals in a population, but if there's no smaller size classes, then we are considering them to be non-viable. Keep these viability classes in mind, A, B, C, and D, because we will be using those color codings coming up in maps and graphs. So what do we know about the conservation status of the western pearl shell in Montana? We do know that they are a declining component of most trout streams across the western part of the state, but even more so on the eastern part of the Continental Divide. We do know that there's only about 20 stream populations that are expected to be viable 50 to 100 years from now. And monitoring it in the last decade found that we had a decrease in populations of over 20% extirpation. In fact, whole watersheds in the eastern part of the state had been extirpated. We do know that the West Slope cutthroat, a declining species as well, is also their primary native host, but they are using other fish species such as brook trout and rainbows. Therefore, what we want to really figure out with this research is why some populations of pearl shells are maintaining viability while others are not. We can see on the left, there's really no recruitment limitation factors here. We have nice cutthroat populations, a lot of adults producing juveniles, whereas the recruitment factors on the right could be the wrong host fish, the non-survival of juvenile mussels as they settle into the benthic. We see the skull and crossbones there. So the three factors we're going to investigate with this research is the host fish presence and densities, the benthic habitat that juveniles have to survive in, as well as the production of glaucidia by those mussels in those populations. Here are our 25 study sites for this project across 5 watersheds, 6 actually, and they encompass a variety of ecoregions, elevational ranges, and we have all viabilities represented within each watershed. If you saw Kristen's talk earlier, she focused on the Big Hole and the Flint Rock watersheds. We put data loggers out to record temperature during the mussel's reproductive period so we can keep track of that. We evaluated habitat data collected at the 25 western pearl shell stream sites to see how this correlated with pearl shell viability. Of course, our hypothesis is that the better stream habitat is going to lead to better mussel populations. We collected percent benthic sediment finds in riffles and pools, as well as channel stabilities using both pebble counts and sediment grid counts shown here. And we will present this with percent fines in the riffles and pools. We evaluated our second hypothesis that pearl shell viability was correlated with reproductive status by determining status, gravidity, and timing of conglutinate release across all 5 watersheds with varying stream temperature regimes, viability status, by revisiting these pearl shell populations multiple times during their known spawning period and evaluating the stage of gravidity that they were in. We can see on the left here a very partially gravid mussel with its gills barely inflated versus a fully gravid marsupial gill here on the right of this female. We did mark individuals, so our revisits, we can track individuals' progression through this developmental stages. And if you saw Kristen's talk, you could see how investigatively in depth she went from egg to Glaucidia development stages. To evaluate our third research hypothesis concerning host fish densities, species susceptibility to Glaucidia, and gill infection rates, once full gravidity of the mussels at a site was documented in a certain percentage of the pearl shell individuals, we fished paired zooplankton nets below the beds. We then electrofish sites 50 meters below and 50 meters above the mussel beds. All salmonids captured during these runs were measured, weighed, and their gills were evaluated under a microscope or lighted magnifying lens to quantify the Glaucidia on their gills. Infections on the gill were rated as low, moderate, or high. We could see that lower fish in the corner there as a very high load of Glaucidia, greater than 50 per side. And these sites were fished on generally just once per season, but some sites did have multiple visits. All right, let's get to some results. These results are our percent riffle grid fines, and these will address our stream habitat, benthic habitat conditions, and viabilities. At first glance, streams with non-viable populations, Ds and Cs, do appear to contain a higher percentage of riffle grid fines. Although this relationship is fairly weak with an R-squared of 0.15, there does seem to be a break point between viable populations and non-viable populations at about 5% riffle fines, and this appears to be limiting the survival of juvenile pearl shell mussels. We were very successful at determining the timing of gravidity and conglutinate release across all sites and both years. We have graphs like this for every site. Across the y-axis is the percent of mussel individuals that were examined that trip, and across the x were the dates. We could see Memorial Day, basically about 50% of that population was 50% gravid, and as we progressed to June, our gravidity increased, and by June 17th, most of the population was fully gravid. Conglutinate release happened shortly after, and then we could see that by July 1st, the majority of that population was non-gravid, having released their conglutinates. We hypothesized that higher gravidity up to 50%, assuming a 50-50 male to female ratio, would be occurring in the more viable populations with dense mussel beds. The percent of pearl shell individuals documented to be partially or fully gravid on each visit is on the y-axis, and viability across the x-axis. A fairly weak, non-significant relationship between gravidity and viability was observed across all watersheds. All populations of viable mussels, whether A through D, exhibited gravidity across many of their individuals. Even some of the D populations, where a few mussels were examined, all showed some signs of gravidity. This graph represents both years of fish shocking data in host fish densities as CPU, catch per unit effort, and our number of salmonids caught per minute of shocking was on the y-axis, and the viability ranks of our pearl shell populations on the x. We see a very decent relationship with higher host fish densities caught in the viable population sites versus the non-viable population sites. As a comparability of methods, the 4 highest CPUE was at Clam Creek, and these were multiple dates with different crews. There are at least 6 visits to D sites on the right-hand side that basically had 0 salmonids caught during those visits. Despite seeing a fairly good relationship between our host fish densities and viable sites, this does not carry over to the percent of salmonids that actually had gill Glaucidia present. We could see on the y-axis the percent of the catch that was infected with Glaucidia and our viable populations on the bottom. We can see that even non-viable populations with fewer fish had some fairly high numbers or percentages infected with Glaucidia. And in fact, that French Creek one in the upper top right corner had just 1 trout caught in the whole reach, but it was infected with Glaucidia. So the relationship between non-viable and viable infection rates is not quite holding up. All right, let's summarize the host fish density and infection rate results. Host fish densities correlated very well with the viability of pearl shell populations. Mussels do need host fish present to be able to recruit. 0 host fish or very few guarantees non-recruitment. But more importantly, we need the proper size range of fish present and the proper species. Juveniles were more important at getting infected than adults for the Glaucidia success. Look at that rainbow trout on the right, just loaded with Glaucidia. That's one successful recruitment event when those baby mussels drop off. Unfortunately, percent of Glaucidia infected fish had a very weak relationship with pearl shell viability, and we reported multiple sites across all viability classes with 0 infected fish near the mussel beds. And finally, A and B viable populations across multiple watersheds are maintaining recruitment by solely using brook trout as their host fish. To conclude, the long-term pearl shell survival remains a concern statewide, with only 20 viable populations persisting, and many of the non-viable populations are teetering on the brink of extirpation in many streams. Hermaphrodism, which has its own problems, may be playing a larger role in why some of our pearl shell populations are successfully gravid every year. Many of our non-viable western pearl shell populations with decent primary host densities have poor benthic habitat, which is limiting juvenile survival. Benthic sedimentation may be responsible for recruitment failure in at least 50% of these non-viable populations. And finally, in streams with high quality benthic habitat like Marshall Creek and the Yak River, even though we got low salmonid densities, these had decent infection rates and were producing juvenile recruitment. So the take home is it doesn't take many heavily infested fish to produce viable western pearl shell juveniles. Thank you, and I'll take any questions.